Hi students, this is Suresh, biology faculty. In the previous session we discussed about the pollination aspect. Now we are going to discuss about the pollen-pistil interaction. So what is meant by pollen-pistil interaction? So pollen-pistil interaction means the stage starting from deposition of pollen grains on stigma and reaching the pollen tube to the ovule. So from pollen grains to stigma and pollen tube to ovule. So these all are called pollen-pistil interactions. So what will happen in the pollen-pistil interactions? In this, the pollen has to identify the same species. So that means if hibiscus pollen grain falls on the stigma of hibiscus only, then it is same, then it is compatible. Suppose it falls on other stigma, then it is incompatible. Suppose if hibiscus pollen grain falls on and Lycopersicum tomato stigma, then it is other species. So the first step is identification of compatible species, whether same species or differences. If same species means it will promote. Otherwise it is different means the pollen tube growth will be inhibited. So how they will interact? How they can identify whether it is compatible or incompatible? By chemical interaction. So by chemical interaction, so they can identify whether it belongs to same species or different species. So in this process, chemicals present on pollen grain and chemicals present on whatever we call the pollen grains present on the chemicals present on pollen grain and the chemicals present on stigma will identify whether it is compatible or not. So when it is compatible, the pollen grain is accepted by stigma, which initiates the next events. So what are the next events? What is? Like the next events means like it is the germination of pollen grain takes place. So germination of pollen grain means where the pollen grains will form a pollen tube so that it reaches to the pollen tube will reach to the ovule. So the next process which happens, that is germination of pollen grains, which takes place. It depends on the compatibility of the stigma. So when they are compatible, then the next process continues. So here many plants prefer outbreeding. Outbreeding means most of the plants prefers cross-pollination than self-pollination. So what is the reason for preferring cross-pollination over self-pollination? The reason is due to continuous self-pollination, it leads to inbreeding depression. So inbreeding depression means the hybrid which is formed, it is less vigor. By self-pollination, the hybrid which is formed, it is less vigor. To overcome this inbreeding depression, so most of the plants will prefer cross-pollination. So how to promote cross-pollination and avoid self-pollination. So in plants, there are different mechanisms to avoid cross-pollination and avoid self-pollination and promote cross-pollination. Like the release of pollen grain and stigma receptivity. Receptivity means stigma is ready for pollination. They are not at same time. So when they are not at same time, so there is no chance of self-pollination. Obviously, it has to go for cross-pollination. When anther and stigma are present at different positions, at different heights, for example, if they are present at different heights, anther is present at this point and stigma is present at this point. So then the chances of self-pollination are lost. They are less, which promote cross-pollination. Self-incompatibility. Self-incompatibility means in some species like dolichos, the stigma of dolichos will not accept the pollen grains from the same flower. That is called self-incompatibility. And in few flowering plants, unisexual flowers are produced. Obviously, it has to go for the cross-pollination and dioecious condition, where the male plant and female plant are present separately, which will promote sure cross-pollination. So here we'll discuss one by one the aspects which we are discussing, like unisexuality. So unisexuality means there will be flowers with either male flowers or female flowers. So there is no bisexual flower. So obviously here there is no chance of autogamy. Only either cynogamy or xenogamy. Dichogamy. In this mechanism, the anther will mature at different timings and female part stigma will mature at different timings of the same flower. Then obviously self-pollination is avoided. Self-sterility. That is, self-sterility means the female reproductive part, that is stigma, will identify the gene which is present in the pollen grain of the same plant, which doesn't allow this. This is called self-sterility, where the self-pollination is avoided and promotes cross-pollination. And you are seeing hercogamy. Okay. So hercogamy means where it is a natural barrier that prevents pollen of same flower to pollinate the ovule of the same flower. Heterostyly. Hetero means styles and stigma are present at different heights, which prevents self-pollination. Okay. And next, pollen prepotency. Pollen prepotency means here when a stigma receives pollen grain of the same flower, from the same flower, as well as the pollen grain from other flower of the same species, the pollen grain of other flower will germinate faster than the pollen grain of the same flower. So it is called pollen prepotency. So where the pollen grain from other flower is promoted, same pollen grain is not slow. The growth of pollen grain is of same flower is slow. So here you can see that the maturation of male and female parts of a flower at different timings. So here if you see protandry, protandry means male matures first in clerodendron, then the stigma. Male means the anther releases the pollen grains before the stigma is ready for receptivity in the same flower. Protogyny. Protogyny means where the female matures first. Female matures first means stigma is ready for receptivity. But the pollen grain of the same flower doesn't release the pollen grains. So obviously it has to go for cross-pollination. Okay. So these are the outbreeding devices which we discussed. Okay. So here in outbreeding devices, by various devices like anther and stigma are present at different position, production of unisexual flowers, okay, where the same male and female flowers doesn't, they are mature at different timings, self-incompatibility, where the gene identifies the pollen of the same plant, where it doesn't allow. So here you can see self-incompatibility. Here the genes present in stigma are S1 and S2. When the pollen grain has S3 and S4, it is allowing germination. See here. The second case the same, but when there is a pollen grain with S1 gene, it is not germinating. Only pollen grain with gene S3 is germinating. And this third case, where the pollen grain with S1 is not germinated, pollen grain with S2 is not germinated. That is here, the recognition of self is due to the S gene. If S gene matches, there is no pollen gene germination. So which is called self-incompatibility. Okay. So now, by overcoming all these barriers, so pollen grain has reached the stigma, it has started germination. So here the germination means, what is meant by germination? Germination means formation of pollen tube. From where this pollen tube is formed? Pollen tube is formed from intine of the pollen grain. So intine of the pollen grain will form pollen tube. This pollen tube will come out through germ pore, which will move through stigma, style, and reaches to ovary. Okay. So the pollen grain will start its journey from stigma, reaches to style, next ovary, next to the ovary. So here, when a pollen grain germinates on the stigma, so whenever the pollen grain falls on the stigma, it has two cells, one smaller cell, vegetative cell, and larger cell, generative cell. Okay. So the generative cell divides, that is, as and when the pollen grain germinates, so the pollen grain has formed a pollen tube. The generative cell will divide and form two male gametes. That is, the two male gametes from generative cell and vegetative cell. That is, the pollen tube will carry three cells initially. So one is vegetative cell and two male gametes. Okay. So here this three cells, they reach to, along with the pollen tube, they reach to the ovary. That is, from stigma, style, ovary, then they enter into ovary. Okay. So by the time they reach to ovule, how they can exactly reach to ovule? So the reaching process here, in the embryo sac, in synergids, there is an apparatus called Filiform Apparatus, which will attract the pollen tube, so that the pollen tube reaches to the ovule exactly. So the Filiform apparatus will secrete chemicals which will attract the pollen tube by secreting chemicals. So this is the function of Filiform apparatus. So pollen tube enters into the embryo sac through micropyle. Here The pollen tube can enter into the ovule either through micropyle, chalaza, or through integuments. But when it comes to embryo sac, the pollen tube enters into embryo sac through only micropyle. The reason is the filiform apparatus present in synergids, which will attract the pollen tube. Okay. So now we have discussed that pollen tube is traveling. Okay. Okay. So this is what process happens during the traveling of the pollen tube. And during plant breeding process, that is in agriculture research institutes, plant breeding programs is carried out to form many varieties in the crops. So during this process, they use some simple steps to carry out a smooth process of plant hybridization. Like the one step they carry out is emasculation. So what is meant by emasculation? This is carried out only in bisexual flowers. This emasculation process is required in only in bisexual flowers. What they will do? They will remove anthers. For what purpose? To prevent self-pollination. To promote the desired pollination and to prevent self-pollination, they will remove the anthers from female pair by using forceps. After removing the anthers from the bisexual flower, the female flower is covered by a butter paper. What is the purpose? The purpose is to prevent the pollination by unwanted pollen. Here what is meant by unwanted? Unwanted means in hybridization program they have a specific female and male plant. Only pollination should take place only between these two. That is, this desired male plant has to pollinate the female plant. So that means the pollen grains from this should fall only on this. That is the reason why they do bagging. Okay. After bagging, when the stigma attains receptivity, so pollen grains are collected artificially from the anther of male plant. With the help of brush, they will collect the pollen grains and dust on stigma. After dusting of on stigma, again they are rebagged and they are allowed to form fruit. That is the process of the pollen germination, the fertilization after that fruits are formed. So this emasculation process is done when it is a female flower. When it is a unisexual flower, they doesn't require any emasculation. Only they require the bagging of flowers. Okay. So here in hybridization program, there is a desired male and female. Only pollination should be carried out between these two so that they can reach to a specific variety. So here they will do emasculation, bagging and rebagging, which are very important steps in the hybridization program, which is done in agriculture research institutes. So here you can see there are major steps in the process of hybridization, like selection of parents. Selection of parents means in a group of plants they will select male and female. Emasculation, if it is a bisexual flower, they will remove the anthers from female. Bagging, that is covering the female flower with the butter paper. Tagging, that is writing the details on the bag, on which date it is emasculation is done, pollination is done, pollination. Collecting F1 seeds, further the next step, again doing the same process for many generations. So this is how the hybridization process is carried out in forming a variety. So here you can see the flow chart of a hybridization program. So the first emasculation is done, bagging, pollen grains are collected, and we open the bag and we dust on the stigma. Again rebagging, immediately it is done rebagging after artificial pollination takes place, then fertilization takes place. Okay. So here, as and when the pollen grain falls on the stigma, whether by artificial means or natural means, either pollen grain reaches to the stigma, it will form the pollen tube. So here you can see the pathway of pollen tube. It has started its journey from stigma, next it has entered into style, next it has entered into ovary, next it will enter into ovule, next it enter into embryo cell. So here from stigma, style, ovary, ovule, it will enter into embryo sac which is present inside the ovule. So here the pollen tube will enter, that is when it is started its journey, the pollen tube has 3 cells, one vegetative cell and 2 male gametes. And when you come to this, that is a pollen tube initially has 3 cells, 2 male gametes and 1 vegetative cell. By the time it reaches to ovule, vegetative cell degenerates. Only it is left out with 2 male gametes, that is called sperm cell. So gradually here the tube nucleus, that is nothing but it becomes very small. Okay. So here the pollen tube will enter into one of the synergy. That is, as synergids has filiform apparatus. In an embryo sac, there are 2 synergids. So one is called degenerating synergid, to which the pollen tube enters. Okay. So the pollen tube enters into the degenerating synergid and releases the 2 male gametes. So now pollen tube has released 2 male gametes into the synergid. So what will happen to these 2 male gametes? So these 2 male gametes, one male gamete will fuse with the egg cell. That is, the male gamete will fuse with egg cell. Hello. Sir, tell me, sir. Fine, sir, I am in the class, sir. Sir, in the live streaming class. Okay. Okay. So here this the pollen tube releases 2 male gametes. So these 2 male gametes will be releasing into the cytoplasm of the synergid. So in this, one male gamete will fuse with the egg cell. So the fusion of haploid male gamete with haploid egg cell, it is called syngamy. Okay. So on syngamy, zygote is formed which develops into embryo. So what happens to other male gamete? So this other male gamete fuses with 2 polar nuclei which are present in central cell. So which fuses to form a primary endosperm cell. So which is a triploid. So here the male gamete is haploid and the 2 polar nuclei are haploid, N plus N. So N plus N plus N that is 3N. It results in the formation of a primary endosperm cell which develops into a nutritive tissue, that is endosperm. So which is called triple fission. So in plants, that is specifically in angiosperms, there are two fusions in the fertilization process. So one is syngamy and another one is triple fusion. In syngamy, the male gamete fuses with egg cell and forms zygote. In the triple fission, the male gamete fuses with two polar nuclei which will form primary endosperm. So which is called double fertilization. So here you can see in the picture very carefully, this is egg cell which is present in the egg apparatus. Okay. So one male gamete is fusing with egg cell which is present in the egg apparatus. So this is one male gamete, haploid plus haploid. It results in the formation of 2N zygote. Other male gamete fuses with two polar nuclei which are present in the central cell. That is, the two polar nuclei which are present in the central cell, they fuse. That is, N plus N plus N. It results in the formation of 3N, that is endosperm. That is, by syngamy, the end product of syngamy is zygote which is diploid. The end product of the that is triple fission, is primary endosperm cell which is primary endosperm nucleus which is called, which forms endosperm, which is triploid. So triple fission, that is syngamy plus triple fission, is called double fertilization. Syngamy plus triple fission is called double fertilization, which is specifically seen in angiosperms. In flowering plants, it is one of the unique features of angiosperms, where double fertilization is seen. So fertilization has completed. What happens after this? After fertilization, the development of embryo takes place. After fertilization, we can see the development of endosperm. Next, development of embryo. Next, seed formation. So here, these are the three activities which takes place after double fertilization. So initially it starts with development of endosperm. So what happens in the endosperm development? By triple fission, there is a primary endosperm cell which is formed, which develops into endosperm. So endosperm is a nutritive tissue which is filled with food materials, which gives nutrition for the embryo. So to development of embryo, initially a little amount of endosperm is required. That's why endosperm development starts first, then later on you can see the embryo. So here the primary endosperm nucleus, which is in short form it is called PAN, divides to form endosperm. So this endosperm acts as a reservoir of food materials in the developing seed. So which gives food materials for the development of embryo development. So for the development of embryo, it requires food materials. From where it will get these food materials is endosperm. So there are different modes of endosperm development. Like in some few plants, we can see in the formation of embryo, initially what happens is the pen, that is primary endosperm, undergoes nuclear divisions only. Only nuclear divisions, no cytokinesis. Then it will form many nuclei. Later on, after formation of many nuclei, cell wall formation takes place. That means initially it is formed with many nuclei, which is called nuclear endosperm. But in some other plants, it is not like this. It is a little bit quite different. As and when nuclei is formed, immediately cell wall is formed. That means immediately the cell wall is formed means we can see immediately the cells. That is, which is called cellular endosperm. Heliobial, which is in between the nuclear and cellular. So it will form transverse wall towards micropylar regions, and it will start with cell wall formation from micropylar and chalazial chambers. Later on, gradually it will stop. So that means in the formation of endosperm, there are nuclear endosperm, cellular endosperm, heliobial endosperm. So here you can see the diagrams where in free nuclear endosperm many nuclei are formed. So these are nuclei. Later on, cell wall is formed. But here in cellular, as and when a nuclear division takes place, immediately the cytokinesis takes place, which leads to formation of many cells, which is called cellular endosperm. When you come to heliobial endosperm, towards the chalazial end, the cell wall formation starts. This here you can see the cell wall formation, which proceeds towards micropylar end, which is called heliobial endosperm. So now endosperm development has started. So endosperm is formed. So it can provide nutrition to the embryo. So after formation of a little amount of endosperm, then we can see embryogeny, that is embryo development. Okay. Embryo formation is similar in monocot and dicot embryo. Okay. So here, first what happens? It starts with zygote. Zygote means which is a diploid unicellular. So zygote is a single cell diploid structure. So this zygote undergoes transverse division. Here you can see one cell of zygote undergoes transverse division. So the lower cell is called basal cell and the upper cell is called terminal cell. So from the basal cell, you can see the suspensor cell. So we can see the suspensor cell. Okay. So here you can see the suspensor cell, which is developed from the what we call from the basal cell. So from the terminal cell, the embryo develops. You can see from one cell 2 has formed, from 2 4 has formed. So many cells like embryo, from the terminal cell embryo develop, from the basal cell we can see development of suspensor. Okay. So here you can see that is embryo develops from terminal cell and basal cell develops into suspensor cell. Okay. So this, whatever the in suspensor, this cell which is present, that is the terminal cell, so which is called hostorium. Okay, the terminal cell is swollen, which is called hostorium. So which will take the nutrients from the endosperm. So in the embryo development, we can see that is there are different stages in embryo development. So initially the embryo is globular shape. Initially the embryo is globular shape. Later on it will be heart shape. Later on it attains a shape that is pro-embryo. Later on it reaches to a mature embryo. That is, in the stages of development of embryo, there are different stages. That is, embryo is globular shaped initially, later on heart shaped, later on pro-embryo, later on mature embryo. So what you are seeing, that is, when we come to this immature embryo, it looks like this in the dicots. So this is a dicot embryo where you are observing different parts which is present inside it. So in this dicot embryo, these two are cotyledons which will store food materials. That is cotyledon 1 and cotyledon 2. And the part which is present between these two cotyledons is plumule, which gives rise to shoot system. And the base, that is radicle, which gives rise to root system, which is covered by a protective structure called root cap. Okay. And the part which is present below the cotyledons, it is called hypocotyle. So it is called hypocotyle. So a dicot embryo has these parts like plumule, radicle, cotyledons, hypocotyle, root cap. These all are present. What about the monocot embryo? So here if you take this monocot embryo, in monocots the embryo has only one cotyledon. The embryo has only one cotyledon. So this single cotyledon is called scutellum. So what happens to other cotyledon? The other cotyledon is reduced. The reduced cotyledon is called epiplast. The reduced cotyledon is called epiplast. So here the cotyledon, in between the cotyledon there is a plumule which gives rise to the shoot system. So it is covered by a protective cap-like structure which is called the protective sheath-like structure, which protects is the coleoptile. So coleoptile is a protective sheath-like structure to plumule. Whereas when you come to radicle, the radicle is protected by a protective pad-like structure that is called coleorhiza. So radicle is protected by coleorhiza, plumule is protected by coleoptile, and the single cotyledon which is present, it is called scutellum. In monocots like grass, wheat, these all are monocots where only one cotyledon is present, which is called scutellum. So here these are the differences between epicotyle and hypocotyle, coleoptile and coleorhiza. Epicotyle means which is present between, that is plumule and cotyledon, which is present above. Okay. So here epicotyle elongates so that cotyledons remain during hypogeal germination. In epigeal germination, hypocotyle elongates so that there. Okay, the terminal end of epicotyle is plumule, the hypocotyle is radicle. The difference between coleoptile and coleorhiza. Coleoptile will protect the shoot apex, whereas radicle and root cap are protected. Okay. Coleoptile has a pore through which first leaf will come, but coleorhiza is not like that, it is a solid structure. It protects plumule, it protects radicle in the soil. Okay. So here, this coleoptile will below beyond the coil, it will stop, that is whenever the below the pore. So these are the differences between the coleoptile and coleorhiza, epicotyle and hypocotyle. So these are the parts present in a seed. So the embryo development takes place inside the seed. So the embryo development takes place inside the seed. So here you can see the seed develops, after fertilization, the seed develops into embryo. After fertilization, the seed develops into, ovule develops into seed, and ovary develops into fruit. So here this, if you see this, the endosperm, which helps in the nutrition of the endosperm, which helps in the development of the nutrition to embryo. Okay. So here you can see that is the development of, after fertilization, ovary develops into fruit, ovule develops into the seed. Inside the seed we can see the embryo, the endosperm. So here there are two types of seeds. One is non-endospermic seed or non-albuminous seed, and another one is endospermic seed. So what is meant by non-endospermic seed? So non-endospermic seed means where the entire endosperm is utilized. It doesn't mean that the non-endospermic seed means, it doesn't mean that endosperm is not formed. Endosperm is formed, but here the entire endosperm is used. When entire endosperm is used, what will happen in a mature embryo? We can see, that is, in a mature embryo we can see only embryo. That is, in a mature seed, in a non-albuminous seed, only we can see embryo, no endosperm. What are the examples for this? Like beans, pea, groundnut, where the endosperm is completely utilized. What about endospermic seeds? Endospermic seeds means where the endosperm is not completely utilized. So in a mature embryo, in a mature seed, we can see both embryo and endosperm. So you can see both, like maize, wheat, barley, castor, sunflower, where the edible part of these seeds is endosperm. So here you can see the differences between non-endospermic and endospermic. So this is non-endospermic seed, where the endosperm is completely utilized. There is no endosperm, only embryo is present. So this is embryo with radicle, plumule, all. But here there is endosperm as well as the embryo with plumule, radicle, etc. Example for this is maize. For non-endospermic is broad bean, groundnut, pea, etc. So in broad bean, groundnut, peas, the edible part is the cotyledons. But here it comes, the edible part is endosperm. The edible part is endosperm. So these are the differences between the albuminous seed and non-albuminous seed. Albuminous means endospermic seed, where endosperm is not completely used, as we can see both embryo and endosperm. But here it is not like that. Endosperm is completely used, where we can see only the embryo, no endosperm. Example pea, bean, mustard. Here example coconut, castor and maize. And when you come to fruits, so till now what we discussed is about fruit, seed. So now we are going to discuss about the fruit. From where this fruit is formed? The fruit is formed from ovary. After fertilization, ovary develops into fruit. Ovary wall develops into fruit wall, which is called pericarp. Ovule into seed. Inner integument into inner seed coat. Outer integument into outer seed coat, testa. Funicle, stalk of the seed. egg, embryo, secondary nucleus, endosperm, antipodal cells, degenerate, synergic cells, degenerate. Nucellus is also completely used, but in some plants it is left out even after fertilization, like coffee seeds. In coffee seeds, we can see even after fertilization there is left out nucellus, that is called perisperm, and hilum we can lick like a scab. So these are the changes which takes place after fertilization. These are called post-fertilization changes. So here you can see the clear diagram where different parts are seen. That is embryo, endosperm, the left out nucellus after fertilization, perisperm, which is seen only in some plants like beet, black pepper, which is diploid. It is diploid, it is triploid, it is diploid, remaining all are diploid. So pericarp means which is the fruit wall of ripened fruit. Okay. So here you can see many fruits. So in fruits there are true fruit and false fruit. What is meant by true fruit? Only ovary will develop into fruit, no other part. But false fruit means along with ovary, other floral parts will develop into fruit. Like apple, where in apple along with thalamus, ovary develops into fruit, which is called false fruit. In apple, the edible part is thalamus. So if you see an apple, so what you are eating, it is thalamus. What in the center will throw out, it is with seeds. This is true fruit. This is thalamus, which we eat. It is called false fruit. And in fruits, there is a concept called parthenocarpy. Parthenocarpy means formation of fruits without fertilization. So without fertilization, we can see that is the formation of fruits in some plants like banana. Okay. And here we can see that is in banana, in some grapes and oranges. The best example for parthenocarpy is banana. Okay. Where in banana, oranges and grapes, where we can see the development of fruits without fertilization. So the fruits which are formed without fertilization, they are seedless fruits. They don't have seeds. Okay. You can see seedless fruits. Nowadays we are getting lot of seedless fruits like seedless grapes, seedless oranges. Even we are getting seedless watermelon, which are parthenocarpic fruits. Okay. So now we are going to discuss about the next aspect in the seed, that is formation of seeds without fertilization. That is development of seed without fertilization. How it is possible? Okay. Here in Epomixes, we can see the embryo develops without fertilization. That means by asexual means. How it is possible? How embryo is formed without fertilization? Here we can see different stages. Like we can see apomictic embryo sac, apomictic embryo. Apomictic means embryo sac is formed without, that is by apomixes. So here embryo sac, haploid apospory means embryo sac develops from haploid megaspore, which is a regular and a normal development which we are seeing. Diploid apospory means embryo sac develops from nucellus. Nucellus means which is diploid, but megaspore means which is haploid. How apomictic embryos are formed? How it is possible? Like in some plant, the embryo develops from male gametes. in some plants we can see other cells of embryo sac develop into embryo. That means the nucellular cells will develop into embryo. Even in some plants we can see the unfertilized embryos. Okay. And next one is polyembryony. So polyembryony means formation of more than 2 embryos. How it is possible? Is it possible in one ovule? How many embryos are formed? Only one embryo is formed. But here more than one embryo is formed in a ovule, then it is called polyembryony. It's generally seen in many plants. Okay. It is most commonly seen in citrus plants, which is discovered by Leeuwenhoek, and the term polyembryony was coined by Brown. And it is also observed in gymnosperms, commonly than angiosperms. So here you can see, generally from one megaspore, if you see the regular development, one megaspore develops into embryo cell. Fertilization takes place. Okay, fertilization is over. The zygote is formed, primary endosperm cell is formed. And inside the seed, only one embryo is formed. So one embryo gives rise to one plant. This is a normal, a conventional development where only one embryo is formed inside one ovule. But in few plants, like some cells of nucellus will enter into embryo sac, which will act like embryos. That means already one embryo is formed by double fertilization, that is syngamy, and from nucellus, cells which enter into embryo sac, they act like embryos. That means here how many are there? One embryo which is formed normally from fertilization process, and another one which is formed from nucellus. So here you can see there are many embryos, that is polyembryony. So in polyembryony, more than one plant is formed. Anyway, one is formed by fertilization, another from other cells like nucellus, which is commonly seen in citrus, where nucellular cells will enter into embryo sac and develop into the embryos. So what is the importance of polyembryony? It is mostly used in plant breeding and horticulture for getting more plants. And even nucellular embryos are free from diseases, and it is used for propagation of plants like citrus and mango. And nucellular seeds provide genetically uniform seedlings which are same to the parent. And they can be used for development of homozygous diploid plants. Even from eggs and synergies, we can develop artificial production of embryos. Okay. So here, what is the difference between polyembryony and Lipomixes? Polyembryony means formation of more than one embryo from in a seed. Lipomixes means formation of seeds without fertilization. In polyembryony, by fertilization only one embryo, but by other means many different. So here no zygote. Seedlings are identical to single zygote but not clones of parent. That means whichever are formed from nucellular cells, they are similar to the parent. It is seen in mango, citrus. Seedlings are genetically identical, also clones. It is seen in Asteraceae grasses. And Epomixis has lot of importance. That is, Formers has lot of uses of Lipomixes, where we can see the formers has to purchase every time seeds from multinational companies with a lot of cost. By Lipomixes, this can be reduced. So here you can see the plant life cycle. So the seeds germinate and form a plant. After plant, the pollen is transferred. Pollen has reached. Pollination, fertilization, development takes place. Seeds are formed from inside the ovary. Ovary develops into fruit. So these are differences between different concepts which we have covered across, like getinogamy and autogamy. Perisperm and pericot. Perisperm means it is remnant of nucellus which is diploid. Pericot means it is fruit wall. Microsporogenesis, it occurs in anther, inside the anther, formation of microspores from microspore mother cells. So here it is difference between getinogamy and xenogamy, where pollination takes place between two flowers of the same plant, two flowers of the different plants. True fruit, only ovary develops into fruit. Here, along with ovary, other parts will develop into fruit, like apple, strawberry. So that's all about sexual reproduction in flowering plants. This is Suresh from SPSVERSITY. Thank you all. We'll meet in the next session.